Have you ever wondered why biological membranes are crucial to life? Let's dive into the fascinating world of these microscopic structures. Biological membranes are not just passive barriers separating the cell from its environment. Far from it. They are dynamic, complex systems that play a pivotal role in controlling what goes in and out of the cell. Think of them as the gatekeepers of the cell, meticulously monitoring and regulating the traffic of molecules. They ensure that essential nutrients are let in while harmful substances are kept out. But their role doesn't stop there. They are also involved in signal transduction, helping cells communicate with each other, and they provide a framework for biochemical activities. These membranes are made up of a lipid bilayer with proteins embedded within, creating a fluid yet sturdy structure. Their composition and function vary depending on the cell and its needs. Indeed, without biological membranes, life as we know it would be impossible. Imagine a drug molecule about to embark on a journey through the cell membrane. Picture this: our protagonist, the drug molecule, stands at the edge of a vast landscape, the cell membrane. This journey is not for the faint-hearted. It's a complex and intricate world, a labyrinth of lipids and proteins that our drug molecule must navigate. The membrane is primarily made up of a phospholipid bilayer. Picture two layers of molecules, each with a head that loves water and two tails that fear it. The heads, or hydrophilic parts, face the outside and inside of the cell, while the tails, the hydrophobic parts, hide away in the middle. But this bilayer is not just a sea of phospholipids. Oh no, it's peppered with proteins. Integral proteins, transmembrane proteins, peripheral proteins, each with their own role to play. Some of these proteins are like gatekeepers, allowing certain substances to pass through, while others are like messengers, communicating signals from outside to inside the cell. Our drug molecule will need to pass through this protein-peppered phospholipid landscape, but how does it do it? Well, it depends on several factors. The size of the drug molecule, its solubility, and its affinity for the lipid bilayer all play a role in its journey. Larger molecules, or those that are not soluble in lipids, may require the help of those gatekeeper proteins to get across. These proteins can ferry molecules across the membrane through a process known as facilitated diffusion. Smaller, lipid-soluble molecules, however, can slip through the phospholipid bilayer without any assistance. And then there's the electrical charge. The cell membrane has an electrically charged environment which can either attract or repel the drug molecule. So with all these variables at play, our drug molecule's journey is a complex one, but no matter how challenging, it's a trip that's worth taking, because once the drug molecule has crossed this membrane, it can get to work. It can interact with its target, trigger a response, and perform its function within the cell. Now that the drug molecule has crossed the membrane, it can perform its function within the cell. But what guides this journey of the drug molecule through the cell membrane? It's a question that has fascinated scientists for years. The answer lies in the unique and complex structure of biological membranes. Biological membranes are not just passive barriers, but active participants in the drug transportation process. They are like gatekeepers, regulating what enters and exits the cell. Comprised of a double layer of lipids with embedded proteins, they create a selective barrier that only allows certain substances to pass through. Now, how do drugs get across this gate? Well, they need the right keys. These keys are the drug's properties: solubility, size, and charge. Let's start with solubility. Drugs need to be both water-soluble and lipid-soluble to make the journey across the cell membrane. Water solubility allows the drug to move through the body's fluids, reaching the cell membrane. Lipid solubility, on the other hand, enables the drug to dissolve into the lipid layer of the cell membrane and pass through it. Next, we have size. The smaller the drug molecule, the easier it is for it to slip through the lipid bilayer. Larger molecules often require the help of carrier proteins to get across. Then there's charge. The cell membrane is selectively permeable and often prefers neutral molecules. Charged particles, whether positive or negative, find it harder to pass through without assistance. But remember, these factors are not working in isolation. They interact with each other and the cell membrane, influencing the drug's journey into the cell. For instance, a drug might be lipid-soluble and small, but have a charge that hinders its movement. It's a delicate dance between the drug and the cell membrane, a dance that is crucial for the drug's effectiveness. Understanding this process is key to developing more effective and targeted medicines. It's clear that the cell membrane is an active participant in drug transportation, not just a passive barrier. And with that, we inch a step closer to unraveling the mysteries of our cellular world. Yet the journey of a drug molecule is not always straightforward. It's a complex dance, a ballet if you will, that involves multiple players and several steps. Let's delve into the different ways drugs can cross the membrane and the factors that govern this intricate process. First, we have passive diffusion, the wallflower of the dance. This method is simple and straightforward. In passive diffusion, drug molecules move from an area of higher concentration to an area of lower concentration, traversing the cell membrane without any external energy. Lipid-soluble or small polar molecules often use this method. Next, there's facilitated diffusion, the tango of drug transportation. It's a bit more complex than passive diffusion. Here, the drug molecules still move from an area of higher concentration to one of lower concentration, but they require the help of a dance partner, a transport protein. This method is often used by larger polar molecules that can't cross the cell membrane on their own. Lastly, we have active transport, the breakdance of drug transportation. This method is the most complex and energy-intensive. Unlike the other two methods, active transport can move drug molecules against the concentration gradient, from an area of lower concentration to one of higher concentration. But this daring maneuver requires energy, usually in the form of ATP, and the assistance of a transport protein. Now, what determines the method a drug molecule will use to cross the membrane? This dance is guided by the characteristics of the drug and the properties of the cell membrane. Factors such as the size, shape, and polarity of the drug molecule, as well as the lipid solubility, play a significant role. The cell membrane's composition, structure, and the presence of specific transport proteins also influence the transportation method. Thus, drug transportation is a complex dance, guided by the characteristics of the drug and the properties of the cell membrane. It's a fascinating world of molecular choreography that ensures our bodies respond correctly to medication. Understanding this process is pivotal to developing effective drug therapies in the future. So, what have we learned about biological membranes and drug transportation? We've journeyed into the intriguing world of biological membranes, those complex barriers that define the boundary of every cell. We've discovered that these membranes are not just static walls, but dynamic structures composed primarily of lipids and proteins that constantly change and adapt to the needs of the cell. We took a deep dive into the structure and function of cell membranes, unraveling how their amphipathic nature allows them to form a stable barrier in an aqueous environment. We've learned that these tiny gatekeepers play a crucial role in maintaining the cell's homeostasis, regulating what enters and exits the cell, and ensuring the cell's survival and function. From there, we embarked on a journey to understand the role biological membranes play in drug transportation. We've seen how drugs are transported across cell membranes through various mechanisms. Some simply diffuse through the lipid bilayer, while others require specific transport proteins. We've also explored the complex dance of drug transportation, where factors such as the drug's size, polarity, and ionization state, as well as the membrane's lipid composition, can dramatically influence how a drug is transported. We've learned that it's not just about getting the drug into the body, but about ensuring it reaches its target site in the cell in the right concentration at the right time. We've delved into the factors governing this transportation, understanding that it's a delicate balance. Too much of a drug can be toxic, too little might be ineffective. The body's pH, temperature, and even genetic factors can influence how drugs are transported and metabolized. Remember, the next time you take a medication, there's a whole world of biological processes making it effective. This microscopic ballet, this intricate dance of molecules is happening right now inside your body, ensuring that every pill, every capsule, every injection does its job. So keep exploring, keep questioning, and keep learning. The world of biology is full of wonders just waiting to be discovered. Subscribe for more because you won't find me later.